Hello. Today in this video we are going to discuss about recombinant DNA technology. Now, what is recombinant DNA technology? Recombinant DNA technology refers to the process of joining DNA molecules from two different sources or species and inserting them into a host organism in order to generate products for human use. This technology involves identification, isolation, and insertion of gene of interest into a vector, such as plasmid, to form a recombinant DNA molecule which can produce large quantities of that gene fragment. There are steps of recombinant DNA technology. Step 1. Identification and isolation of gene of interest or a DNA fragment. Step 2. Insert the isolated gene into a suitable vector. Step 3. Introduction of the vector into a host. Step 4. Selection of transformed host cell. And the final step or step 5 is multiplication of the introduced gene into the host. Talking about step 1, we can get the gene of interest from various resources. It can be from genomic DNA library, cDNA library, chemical synthesis, or amplification of the desired gene using techniques like polymerase chain reaction. Now for an example, we will take genomic DNA library. From genomic DNA library, we will collect human insulin gene. We will introduce human insulin gene in bacteria. Talking about step 2, insert the isolated gene into a suitable vector. Now we are taking plasmid as a vector. Plasmid is an extrachromosomal circular DNA which is present in the bacteria. We are taking E. coli bacterium, and from E. coli bacterium we are going to isolate plasmid, and we have our gene of interest, that is human insulin gene. Here we need to cut the plasmid using restriction enzyme. A restriction enzyme is an enzyme that recognizes specific short nucleotide sequence and cuts the DNA only at specific site or restriction site. We need to assimilate the insulin gene into the vector. After incorporation of the insulin gene into vector, it is ligated or it is joined using the enzyme ligase. Now we have the molecule called recombinant DNA molecule. Recombinant DNA molecule is the DNA molecules from two different sources or species. Step 3. There are various processes through which we can introduce the vector into a host. They are microinjection, electroporation, liposuction, or direct transfer of a DNA. Chemically, we can also transfer it by PEG-mediated, calcium chloride-mediated, or dextrin-mediated gene transfer. We have our human insulin gene incorporated into the vector, and we need to introduce this gene into a host. This host can be a bacterial cell such as E. coli. After the introduction of the vector and the gene of interest into bacterial cell, we have a genetically modified E. coli with our insulin gene. This process is known as transformation. Next step is step 4. After the process of transformation, we will get 3 types of colonies. First, transformed bacterial cell. transformed bacterial cell with unchanged vector. And the final colony is transformed bacterial cell with recombinant vector. Now we need to select this final colony. There are ways by which we can select this colony, such as antibiotic resistance in a selective medium, colony hybridization. This gene may code for some visible characters. It may be done by blotting test, etc. Now our final step is multiplication of the introduced gene in the host. As we know, we need to make many copies of the gene fragment. So we have the genetically modified insulin gene and the plasmids, which replicate itself inside the host, and thousands of copies of that particular gene will be made by the bacteria. After reproduction, bacteria will divide and increase its number. Now we have our gene of interest in many copies. The gene of interest, or we can say the foreign gene, should be present in the newly produced bacterial cell. The newly produced bacterial cell will contain these foreign genes. If the bacteria contains the foreign gene, we will call that a recombinant bacteria. And this recombinant bacteria is produced by the technique called recombinant DNA technology. Hope this video will help you. Thank you.